So this is a little talk based on some questions I've had from members of our own team that made me realize that there are a couple of things that may be worth discussing in more depth and record that for future people joining the team. So this is an example of colony counts on a TBX media where E. coli colonies can be seen as bluish color due to a particular enzyme E. coli has. Now, you probably all have seen plates like this. And the question is, what does it actually mean if we count a colony forming unit? So I want to explain a little bit what a colony forming unit means. And here's one example. You can see a cell of E. coli under the microscope, which is actually in the process of being lysed by a T4-like phage. So if you're plating out this cell here at time 0, you're plating out that cell, will this cell form a colony on the plate or not? And that's a question I want you to think about for a few seconds before I answer it. And the answer is, of course, this cell is going to die and will not form a colony on the plate. So any cell that is currently infected by a phage or predator or injured in some way and unable to survive and grow will not form a colony on a plate. So we're only counting cells that are viable and not infected by a phage, for example. Now, the next example I want you to think about is we have a cell here that is, as you can see, dividing, forming 2 daughter cells here that are still connected, and then the 2 daughter cells become disconnected. So if we are plating a cell, if we are plating this at this point in time, how many colony forming units are we going to get? And if you're plating out at this time, how many colony forming units are we going to get? So have a think about this for a few seconds before I explain the answer. So if, as long as the 2 dividing cells are not completely divided and held still connected, they will only form 1 colony. So this is 1 colony forming unit. Once the cells are sufficiently separated that during vortexing or mixing, these cells will become completely separated, they will form 2 colonies. So a just dividing cell will only form 1 colony. And here, just to show you that not all cells look like E. coli, this is hypermicrobium, a cell that has a kind of appendage from which the daughter cell is budding off. Just to show you that not all cells look like E. coli. Now this construct here, mother cell, the prostica, the appendage, and the daughter cell budding off, this is only 1 colony forming unit. And these cells remain connected for some time, forming another bud and so on. And this will all be 1 colony forming unit. Some more examples. So here we have streptococci. They form chains of cells, as you can see here in a gram stain. This is a chain of many cells. But as you probably now know already, this chain here, because these cells are connected, is going to form only 1 colony. So this is 1 colony forming unit, but it's many individual cells which are not, I mean, they can individually die, for example, but they are physically connected, so it's 1 colony forming unit. Similar for staphylococci, who are also forming not chains, but these grape-like clusters of cells. As long as the cells are in a cluster, one cluster is only one colony forming unit. So again, many cells can become just one colony forming unit. Here, maybe a more extreme example is a filament, in this case of cyanobacterium. Many cells in a filament. One filament is only one colony forming unit. So we have to remember when we are counting colonies that these can represent just a single cell has grown into a colony, or many different cells have grown into a colony. So let's try to define a colony forming unit, which is one viable cell or cluster of viable cells, where at least one of the cells is viable in that cluster, able to grow on the particular medium that you're using under the particular conditions that you're setting into a colony that's large enough so you can see it and count it. So lots of conditions here. So what's most important to remember is that it can be a single cell or very many that are clustered together in some way. You will only know that if you're looking under a microscope. And you can separate cells in a cluster by either intense vortexing and or sonication, or combination of vortexing and sonication. You will probably know that ultrasound can be used to disintegrate cells or live cells completely, but it's a question of the frequency and power and length of treatment, whether you are separating cells or killing the cells. So at a suitable frequency and power, you can disperse clusters without actually killing the cells. That's possible, but it has to be tested. If your viable count, if your colony forming units are increasing due to the treatment, then you're not killing them, but separating them. Another thing that I realized is often not clear is the fact that many bacteria from an environmental sample will not be growing on one particular medium under one particular set of conditions. So most of the bacteria you could count in a microscope. This could be because the cells have different nutritional requirements and provided in the medium, or they may need a partner to grow, or they may simply be sleeping or dormant, or maybe anaerobes, and you have aerobic incubation, or phototrophs or oligotrophs, which are adapted to very low nutrient concentrations, and you're using a rich medium, then these cells are not going to grow. So what's important to remember is that typically only a very small proportion of the total count of cells in the microscope is going to grow on a particular medium. And that's been known as a great plate count, normally coined, given this name by Staley and Knopf in 1985. And they kind of put up this general rule, but of course it's only a rule that only up to 1% of the total direct count from aquatic habitats will grow on a particular plate. One thing that has been observed is when you dilute the medium, so if you take a rich medium and you dilute it tenfold or maybe more than tenfold, you actually get a higher count. That's often been shown, and actually that's something we found ourselves as well. The reason for that is primarily that many bacteria in the environment are adapted to grow under low nutrient conditions. They have high affinity uptake systems. So when you give them lots of substrate or lots of phosphate, they just pump themselves full and die. It's called substrate accelerated death. Another thing worth knowing is that if you autoclave agar with phosphate buffer, it generates toxic hydrogen peroxide. That has been shown by a Japanese group quite conclusively. You get a higher count from environmental samples if you're autoclaving the phosphate separately. Also, agar can contain inhibitors of growth, so washing the agar can increase your plate count. Here just a few examples from a paper looking at bacteria in a eutrophic lake in northern Germany, where you can see the effect of different buffers. Here is the culturability, so the percent of the total count that actually grows on the plate, which in the control is higher than 1%, just a little bit higher than 1%, but with using of phosphate, the plate count goes down, and the best plate count you get with a non-phosphate buffer like keepers. Another example, different media. They often use R to A medium, gives you a lower plate count, and more media that are aiming to mimic the natural environment, like this synthetic freshwater medium here gives you a higher plate count. So it depends on many things, including the medium. And phosphate is in higher concentrations actually killing a lot of oligotrophs. So don't be surprised if you don't have a large number of counts on your sample when you're expecting the sample to be full of bacteria. Most of the bacteria in the sample will not be growing on a particular plate. Now we're moving to some statistics of plate counts, and I have to introduce Poisson statistics, which is relevant for counts of events. So that is when you can count how many times something has happened, but you can't count how many times something did not happen when the non-events are not countable. And that means there is no maximum count. Because you can only count events and not non-events. You can't calculate a proportion, which if you have a proportion, you could use binomial statistics. But when you have when each marble you pick from an urn is either one color or another color, then you can calculate a proportion and use binomial distribution. So Poisson distribution is about counts and not proportions. So some examples: the number of radioactive decays per time interval. So you can't count the non-decays, you can only count the decays. Number of cells in a particular volume, number of virus particles in a particular cell, number of cells passing a detector in a flow cytometer or culture counter, number of cells in a grid of a counting chamber, or number of colonies on the plate, or a number of raindrops on flagstones, so per area. So it's always a count of something per either time interval or length interval, or per area or per volume. So if you think about this in terms of the generating process, this is a very, very simple process where you basically have the constant probability of an event occurring per unit time or unit area or volume. So this is a very simple process, really. And that means that the events are independent. One event does not influence the other. And I'll show you an example on the next slide of simulated radioactive decay. So here are the events over time. They occur randomly, but the probability of them occurring per time interval is constant. So if you then overlay this with a grid, you can then count the number of events per interval. So 0, 0, 3, 1, 0, and so on, and plot that down here. So this is the number of events per interval, 0, 0, 0, 3, 1, 0, and so on. And you do that many times, and then you can generate a plot of the probability or frequency of an event for 0 events or 1 event per interval, or 2 events or 3 events per interval, and so on. And these are the simulated data here, the crosses, and fitted to a Poisson distribution. These are the circles. So at 200 events, the fit to the distribution is already quite good. But with the example here, that's just, I think that was 10 counts. The fit to the distribution is likely not so very good, but obviously partly due to chance. Just to show you briefly the equation of the Poisson distribution. What's more important is to think about what the shapes actually look like. I'll show that in a second. The Poisson distribution has only one parameter, which is this mu here, which is the mean. K is a number of events, so that's not a parameter of the distribution. And so this is the mean, either the population or true mean or the sample mean. And the variance, this is a unique fact about the Poisson distribution. The variance is equal to the mean. In other words, the standard deviation is equal to the square root of the mean. So there is these two parameters, which, for example, in the normal distribution are independent. You can characterize a normal distribution with a mean and a standard deviation. But here in the Poisson distribution, these two parameters are not independent. It's just that means it just has one parameter. Okay, so just to show you the kind of shapes this distribution can take on, can be very skewed or fairly symmetric. We have different means here. The mean for the black distribution is 0.5, then red is 1, green is 2, blue is 4. So you can see here rapid decline of frequency with a number of events. If the mean is below 1, if it's 1, the first two frequencies are equal and then it drops off quickly. For 2, it first increases, stays constant, goes down again, and then for 4 it becomes a little bit more symmetric already. So the shape of the distribution can change a lot when you're changing the one parameter the distribution has, the mean. Okay, now what that means is that the more counts you have, the better it is, or the lower the error is. So I made some more plots to show that. This is because of the relationship that the standard deviation is equal to the square root of the mean, which means that the relative standard deviation, also known as a coefficient of variation, which is the standard deviation divided by the mean. So this is the relative standard deviation or coefficient of variation. That is now the square root of m divided by m, which you can rearrange write in different ways. So 1 over the square root of m. If you're plotting that, it looks like this. So our relative standard deviation is very high if you have a count of 1, and then it drops quickly, but you still have quite a large error if you have a count of 30, 50 or so, and it only decreases quite slowly. So there's not much of a difference between 100 and 200, for example. You're zooming in up to 30, you can see if you have a single colony on your plates, the relative standard deviation is 100%. So you have a very high so-called sampling error. And this error is unavoidable. And the key thing to realize is the higher the count, the lower your relative standard deviation. So this is why it's good to have a higher count. The higher the better from that point of view, although the benefits are of the increased number of counts are diminishing. So it would be great to have lots of colonies on your plate, but then we have another problem that the colonies might be overlapping, so you can't really distinguish them and count them. So here you can see in some cases the colonies are here. Here you can see the two colonies just about recognize that they are separate colonies and count them as two. Here they become so close, the two colonies, that are hard to distinguish. And if the two cells originally on the plate were too close together, they will only form one colony. So you will get this coincidence issue that cells too close together will form one colony or two colonies that are hard to distinguish, even if you're very careful when you're counting. So this is why a too high number of colonies on the plate is not good. But having many counts on different plates, that's fine, that's great. So we have the problem of overlapping colonies if there are too many. And actually, this is called coincidence. How many is too many depends on the size of the colony, as you can probably see from this picture here from Madhusri. There are also some very small colonies, and they are easier to count or distinguish as separate from the others. So it depends on the size of the colony. So the standard rule that you might see in the literature is to disregard plates with more than 200. But that's based on original work done with counting bacteria in milk. We're using a rich medium and the colonies are going to be large. If you have small colonies, you can count more than 200 without an issue. One trick to do that is actually having submerged colonies. They are much smaller. And you can do this with poor plates or better with layered plates, making sure that all colonies are submerged by having another layer of agar on top. That keeps the colony smaller, so you can count a much larger number in a single plate, and you have a better dynamic range for your count. So you can count somewhere between 30 and 1,000 colonies in a plate without coincidence problems. And there is even the possibility of correcting for coincidence. There is an equation in the chapter by Koch 2007, which I will show you later. I'm not going to go into the details here, but just so you're aware that you can actually correct for coincidence if it's not too large. Remember that the error, our coefficient of variation or relative standard deviation, depends on the total number of colonies counted, not on the number of colonies on a single plate, but all the plates you've counted. That will determine your sampling error. So the other problem is if you don't have enough colonies on a plate. Again, the standard rule is to disregard plates with fewer than 20 colonies, or sometimes you will find other numbers, 25 or 30. And that rule makes a lot of sense if you're a quality control lab, for example, where you can do it again. Or you can optimize a protocol to avoid this problem in the future by changing the dilutions you're plating out. If you're doing routine work, you can optimize a protocol and make sure that the number of colonies on your plates are in a suitable range. But I think we're in a different position. We are looking at environmental samples, and you can never take the same sample again. And ignoring a count, if it's your only count, ignoring that count because it's less than 20 would be foolish in my view, because it's better to have an inaccurate figure of colony forming units and no figure at all. You have to be aware, of course, that if you have a small number of colonies only, you will have a large error. So you have to be careful with interpretation, but better a number with a large error than no number at all, especially if you recognize that the error means you need to be careful with your interpretation, and maybe your number is just an order of magnitude estimate and not more than that. The rule of disregarding plates with fewer than 20 colonies should not be confused with the limit of detection, which is obviously just a single colony is your limit of detection if you have a single colony on all your plates. And if you have that, let's suppose you have no colony at all on any of your plates and you've plated out a certain volume, you can report this as that the count is below the count you would get if there were a single colony on all those plates. So your count is clearly below this number. You don't know how far below, but it's below this number of 1 CFU per total volume counted. That will become a little clearer on the next few slides. So, but if you have just one count or so, it's not, as you know, the error is large. For one count, it's 100%. So you have to be careful with interpretation, but don't ignore this. Okay, here's an example of how it's generally done. A simple example, obviously. So you plated out 0.1 milliliter aliquots of a 1 to 10 dilution and a 1 to 100 dilution of a particular culture on 2 plates each. And the results are you got 200 and 180 colonies and 22 and 16 colonies on the higher dilution. So since there are not too many colonies on the 1 to 10 dilution, you can count all the plates. Because the count here is low, you would disregard the counts from the higher dilution, just count the 1 to 10 dilution and ignore the 1 to 100 dilution, despite the fact that this contains information. There are counts, but we're ignoring them. That's the standard procedure. And then you calculate the CFU per milliliter from the 2 plates from the 1 to 10 dilution, and you work out the CFU per milliliter. Then you can calculate the average of these, and you can calculate the standard deviation using Gaussian statistics. That's how it's usually done. But you have only 2 replicates, so your estimate of the standard deviation will be very will have a high error. Okay, here's another way of doing it that I think is preferable. So the example is the same, the numbers are the same, but because the higher the number of counts, the lower the sampling error, we will now include all the counts. So what we do is we sum all the counts from all 4 plates, which is a total count of 418. And then we sum all the volumes that have been counted. So we have counted 4 plates, and now we need to work out the volume of the undiluted sample that has been plated out. So if you're plating out 0.1 milliliter of a 10-fold dilution, you're actually plating out 0.01 milliliter from the original sample. So 0.01 milliliter of the original sample 2 times, because we have 2 plates, and then 2 plates with a higher dilution, which is 0.001 milliliters of the original sample. So we've plated out 0.022 milliliter of the original sample in this example. And now we can calculate the CFU per milliliter by dividing 418 CFU by 0.022 milliliters, and you get this result. It happens to be the same in this particular example as the other method. But now you can calculate the standard deviation of this from the Poisson distribution as the square root of the total count, which gives 20 or thereabouts. And you can then calculate the standard deviation in CFU per milliliter by dividing that standard deviation by the volume that you've counted, same as above. So you get 929 CFU per milliliter. But remember, this number is not that precise, as it's based on a count of 418. So we can then express the results in this way: 1.9 plus minus 0.09. And I'm not giving any more significant figures here. I was wondering whether I should introduce and explain the concept of significant figures, but I thought it's going to take too long, so I'm leaving that out today. But please, if you're not sure what significant figures are, it's good to know about that. So if you're comparing the results with the other procedure, you see that the mean in this case is actually the same, not necessarily, but in this case, the error is higher from the other method. Again, that depends. Remember, if you're publishing figures like this, don't give too many significant figures. We have only had a count of 118. We have a standard deviation of about 0.1. So it's not justified to give more than two significant figures. And that's very rarely the case, that you have more than two significant figures. Often, in some cases, maybe less than two. So a couple of remarks. Remarks and additions. So we should include counts from all dilutions as long as they are countable, so the plate isn't too full. So also include plates that have a low count, because it has some information, and let's use all the information we can. And if you were to average plate counts from the same sample from different plates with different counts, you're basically giving both plates the same weight, although the plate with a larger count has a lower error than the plate with a lower number of colonies. So just taking averages between plates means you're not considering that the plates with a higher count have a lower error. Another, this is a very, I hope that isn't confusing, a very different example is when we are averaging across different samples. So let's say you take different, you take a sample from the same place at different times and you want to average those, or you take samples from different places in the lake and you want to average over those to give the average count of bacteria in the lake or average count over time. Then if your counts are varying a lot between the different samples, it might be better to use a geometric mean than the arithmetic mean if your data ranges over several orders of magnitude. So here's a simple example, made up, of course, where we have a count of 1, 10, or 100 CFU per milliliter at different times. So a wide range. So the count changes a lot. If you're taking the geometric mean of this, which is the product of the individual CFU values, so 1 times 10 times 100, and then taking the nth root of this, we get 10 CFU per milliliter, which is exactly the middle value, if you like. If you're doing the arithmetic mean, in this case, then we are summing all the numbers here, 1 plus 10 plus 100. So this one here is basically has no effect on the arithmetic mean. The 10 has a small effect on the arithmetic mean. You're basically dividing the largest number by 3. So you get a very different result if you're taking the arithmetic mean and the geometric mean. The arithmetic mean being strongly influenced by the largest number if your data ranges over several orders of magnitude. Actually, many people take the log values of CFU counts and then averaging the log values. If you're averaging log values, what you're actually doing is you're taking the geometric mean of the counts, because the arithmetic mean of the log of the counts is the same as the geometric mean of the counts itself. So I just want you to be aware that averaging log counts and averaging counts is a very different thing. And if your counts vary over orders of magnitude, averaging the log counts is actually a sensible thing to do. So the arithmetic average of the log of the counts is a sensible thing to do. Just be aware that this is the same as a geometric mean of the actual counts. So another thing that I guess should be discussed here is that we have talked about the sampling error so far, but this is not the only error. So we have the sampling error, which is unavoidable. There's nothing you can do about it. This is caused by the cells being in a random location, because you have mixed your sample, obviously, before doing the dilutions, and you're mixing the dilutions while you're doing them. So the cells are in a random location when you're sampling them, and that means you will have the Poisson distributed error from this sampling, where the error is given by this equation, the square root of the mean. But you also have technical errors, of course. You have pipetting errors, counting errors, and these are to some extent also unavoidable, but they can be minimized by good practice. And to estimate the technical errors, you really need to do a lot of independent dilutions, completely independent dilutions, not just different plates, but independent dilution series from the same sample, then use Gaussian statistics to estimate the technical error. And if the technical error is larger than the sampling error, you have to, of course, you should use many independent dilutions and Gaussian statistics to estimate the error. Or better, reduce the technical error. If you have different sources of error, you can check again the reference by Koch for the rules for error propagation, which I don't have time to go into, of how to combine different errors that enter into the same measurement. Just one important thing to remember is that you can always test the procedures. In this case, you can test it by spiking your sample with a known number of viable bacteria that you can somehow distinguish. Maybe they carry GFP or whatever, and you can pick that up. And that's effectively using an internal standard that can test your procedure. So there are two references. If you want to go into more detail or read it up in your own time, the book chapter by Arthur Koch in the big manual Methods for General Molecular Bacteriology, which is this thing here. I hope you can see that. This is one chapter in that book. And the other book is another old book that I got from a second hand from the library that chucked it out. Meinel and Meinel, Theory and Practice of Experimental Bacteriology. I find that the older books actually give much more detail on the statistics and the quantitative side of things. In newer books, you don't tend to find these kind of things, unfortunately. So you have to go back to the older literature. So to summarize what I was trying to explain, a single CFU can come from a single viable cell or cluster of many viable cells. And most environmental bacteria in your sample will not form a colony on one particular medium, even if it's not a selective medium. And that's known as a great plate count anomaly. It's a typical idea that the number of cells that grow is 1% or less. Third point I was aiming to make is that counts follow the Poisson distribution, which means that the higher your count, the lower your sampling error is, which means you should have enough counts to get good results. More than 100 would be good. And you should include all the plates that can be counted and divide by the sum of all the volumes that you've counted. Be aware that a small count will have unavoidably a high error. And you can calculate that. And then you also have technical errors that you need to consider if they are higher than the counting, the sampling error. If you have a very small number of counts, the sampling error will certainly be higher than your technical error. Only if you have a large count might the technical error become as large or larger than the sampling error. We have time for questions. I apologize that there was a lot of text on the slides, but I wanted to make sure that if you read the slides without me talking, it still makes sense and you don't rely on the spoken word. So if you do have any questions, you can ask them. If you have any suggestions as well, everything is welcome. Use a chat or use just unmute yourself and fire away.
Yeah, can I ask a question or get your thoughts about a couple of things?
Okay.
I was really interested to listen to the stuff about the CFU, just as someone who uses CFU data all the time, and maybe doesn't bear that much thought to how it's collected, or well, often it's not reported how it's enumerated. So you kind of make the assumption that best practice is being followed, I guess. But what was interesting to me was, when you're trying to estimate, say, risk of disease from an exposure to an organism, you use like the dose response relationships, which are usually reported as like numbers of organisms being exposed to some sort of outcome. But when you look at the preceding sort of exposure pathway, the environmental compartments that an organism is going through before it reaches the exposure, they're usually reported as CFU. And usually we when doing sort of risk estimates, just assume that the error in the dose response is pretty much as big as the error in the plate counts, and therefore we can assume one CFU equals one organism. Or if we want to be more conservative, we might think one CFU might equal 4 or 5 organisms. But it's a bit, I finger in the wind maybe. And I was wondering, on your opinion on whether there's any way to make those calculations more representative or more accurate, or whether, because all enumerations of organisms are prone to these sorts of errors, that there's nothing much more you could do if trying to estimate final risk from an exposure. If you see what I'm trying to get at.
Yeah, so it's the question with many different aspects. So on the positive side, if we are talking about pathogens, they are generally easy to grow on rich media. So in contrast to many environmental bacteria that I've called oligotrophs, so bacteria that are adapted to grow at low nutrient concentrations, this is not your typical pathogen. So the typical pathogen is much easier to count on a plate. And E. coli as an example, E. coli is one of those organisms which is very difficult to not grow.
Okay.
I mean, you have to be extremely harsh to them that they won't grow. They grow in phosphate buffer media. They grow in rich media. They grow under aerobic conditions, under anaerobic conditions. They grow, and that's why they are so used in the lab, because E. coli, everyone can grow E. coli. Growing environmental bacteria that have specific requirements or are more difficult to grow, that's something you have to learn, and not everyone can. Or, for example, it took me 3 months to learn to grow the bacterium that I was using in my PhD.
Okay.
And we never had enough time in any student project for the students to learn to grow this bacterium. So I could basically not do any student projects with this bacterium. But many pathogens are easy to grow, so that problem is less of a problem. In terms of, however, also E. coli, for example, or other pathogens may be in a resting state, and may have difficulties to recover. So there may always be more bacteria that are not going to form a colony. But here I think we also we have the advantage that a cell that's injured or not viable in some way is less likely to actually cause an infection. Okay. So being able to grow on a plate and being able to cause an infection is kind of the same thing.
Okay. Yeah.
So I think the problem is not as bad as you might think. In terms of the problem of CFU counts and how it's done, if people don't report how they do it, I would generally assume that they do it in what I think is a normal way, just based on my experience with many labs and many people, how they are doing it. And this is just excluding plates with a high number of colonies, excluding plates with a low number of colonies, and just counting the plates that are in the range of 20 to 200 or so. So these are the standard rules that are used in quality control labs. And they make sense if you are in a quality control lab where you can optimize your method for a particular type of sample, because you always count milk. And then obviously you would be a bad scientist if you can't optimize your method for that particular purpose. But for environmental samples, you don't really know what the conditions are, and your method may not be suitable. And you have a mixture of many different bacteria in the sample. That's a very different thing.
Yeah, because often you literally do just get the CFU per mil or per whatever unit volume reported. You don't get the number of colonies that came from reported. So you're not sure, I suppose, how reliable the data are. But I guess if most people would follow this sort of rule of thumb between sort of 30 and 200, then
I guess many people do that. But you never know, depending, if they don't report the details, then probably they follow the normal procedure. That is what would be my suspicion. I generally would regard CFUs more as an order of magnitude estimate than anything more precise.
Yeah. Okay.
Unless the method section suggests that they've spent a lot of effort on the counting. And if they spend a lot of effort on the counting, they probably spend a lot of effort on writing up about the counting. So I think there is a correlation. The less effort you spend about writing up the counting, the probably the less thought you've given it.
Yeah. Okay. Yeah.
But that's just my personal view.
Okay, brilliant. No, thanks, Jan. That's some helpful stuff there. Thank you.